Good evening, RIT Tigers, and welcome to this evening's webinar. We are happy to have you join us today and hope that you and your entire Tiger family are well. Before I turn over the program to our moderator, Professor Maureen Ferran, I wanted to go over a few housekeeping items. Special thanks to our live captionist with us this evening. Thank you for being here tonight. Zoom webinar platform attendees will not be able to interact via video or audio. Only panelists will be broadcasting via video and audio. The webinar will be recorded. Attendees are welcome to interact throughout the program through the Q&A functionality located in the bottom of the toolbar throughout this webinar. We will address questions at the end of the program. You can select your viewing preferences in the upper right-hand corner of the screen. For best viewing, I would recommend selecting side-by-side gallery. With that, I'll turn it over to our moderator, RIT College of Science Associate Professor Maureen Ferran, to begin this evening's program. Take it away, Maureen.
Thank you very much, Bill. It's really great to see everybody. I'm extremely excited about this webinar. I was really excited when I was contacted to help facilitate it. And of course, it's always a marvelous pleasure to see some former students and alumni and get to catch up with them. This is particularly exciting for me because they happen to be three students who worked in my lab for a period of time, and I certainly had the privilege of teaching them in numerous classes. So it's great fun to see them back. I'm sort of the resident mammalian virologist here at RIT. We also have Dr. Julie Thomas, who works on bacteriophage, which are viruses that infect bacteria. And so my research interest for a long time has really been how mammalian viruses infect cells and how they then bypass our immune response. And our immune response can kind of be broken down into two branches, the immediate or innate, or the later or cellular. And I'm particularly interested in how viruses very, very rapidly turn off our ability to defend ourselves. And this really leaves us susceptible. And so if that system worked every single time, we'd never get a viral infection. And so we certainly know that that's not the case. And so that's really my main research interest. I've also sort of branched off in collaboration with other people more recently, but that's still my main focus. And so I'm really excited tonight that I get to introduce you to some of our alumni and former students. And again, the real overall idea of this webinar was to allow both alumni and current students to hear a little bit about what former RIT students are doing in the field working on COVID and SARS-CoV-2. And I was quite surprised that I actually have something like 9 to 10 former lab members out there working actively on COVID. I was quite surprised and pleased to see that so many RIT people are out there really trying to help us manage this pandemic. And so we thought that you could introduce a little bit about some of your research, your goals, and certainly I think for the current students, they'd like to probably later on at least hear about your experience, how you got involved in undergraduate research, and then also how that tied in to your experience after leaving RIT. So I'm really excited. I'm gonna just introduce the former students based on the little itinerary we came up with, no other order than that. Dr. Alec Freyn graduated from RIT. They all got their degree in biotechnology and molecular bioscience. Alec graduated in 2016, and he then went off to Mount Sinai and got his PhD in virology. And he is now at Moderna as a scientist. And I think he's gonna tell us a little bit about the mRNA vaccines and some of the really interesting things that he's been involved with there. Then we have Katie Marquis. She is now a graduate student at the University of Pennsylvania in the Bushman lab. She is primarily interested in HIV, but she also, like the rest of the world, stepped aside at the beginning of the pandemic to contribute and help us manage this pandemic early on. And then we have Callie Donahue, who, Katie graduated in 2015, and Callie graduated in 2018. She's our newest graduate, and she's also a PhD student in virology at Boston University. So I'm really excited to have you all here. And we thought that perhaps we could start off our question and answer section with Alec perhaps describing a little bit about, we've heard a lot about the spike protein of this virus and how important that is for vaccine design. And something I thought you could really speak to that would be very, very interesting also is this idea of mRNA vaccine technology being brand new. And I think that most of us are aware that that's been actually in the works for a couple of decades, approximately. So I thought maybe you could introduce us a little bit to the virus and a little bit about what you're doing with vaccination.
Yeah, thank you very much. Thanks for the introduction. It's great to be here. So I guess I'm brand new at Moderna, so I started just a few months ago, and I think I have to say something like my views are personal, some disclaimer like that. Anyway, so in my thesis work, I was working with mRNA vaccines and got a little bit deeper into the history of mRNA vaccines. So you may have heard in the news that these vaccines are brand new, and we just made them up and developed them in a year, and now we have this really incredible vaccine product. But really what's been going on behind the scenes is a lot of trial and tribulation and many years and many careers of dedicated work, lots of failure, lots of trial and error. So I worked with actually a group at the University of Pennsylvania during my Ph.D., so where Katie is now, Drew Weissman's lab with a professor there named Norbert Pardee. And so Norbert was really spearheading, he's a fantastic scientist. He was really spearheading this mRNA. The problem at the time was how to get mRNA into cells. So I guess a real basic, what the vaccine basically does is you have a piece of the virus encoded onto this molecule mRNA. And the trick is getting it into cells so that they can make that piece of the virus so that you can warn your immune system that the virus, when the virus does come, you already have some knowledge of it. And Norbert tried for years and years and years, different techniques, all he could think about. And I think after 4 years of trial and error, so a whole undergraduate career, he finally was able to partner with a company that made these things called lipid nanoparticles, which are basically little tiny bubbles of fat that you can wrap around the mRNA. And what this does is it basically allows a vehicle to get the mRNA into cells. And so this was actually roughly a decade ago that this discovery was made. And this really spawned the companies Moderna and BioNTech, who now has partnered with Pfizer to make an incredible vaccine as well. And just another kind of side story in terms of the coronavirus spike is the way that we were able to design the coronavirus spike for this vaccine leveraged work mostly out of the Vaccine Research Center, which is a government body under the National Institute of Health. And there's a laboratory there run by Barney Graham, in which they look at these different virus families that have pandemic potential, and they're able to make modifications to different regions of the virus to make them better vaccine candidates. And so this program has been going on for many years. And so when the coronavirus came out as a pandemic, a virus of pandemic potential, Barney looked into his research, found that there were ways that we could tweak this new virus to make it a better vaccine candidate, and then openly shared that research with the world. So that's kind of been a bit of the journey of what's gone on in the past 10 years that has led to such an amazing vaccine coming out.
That's awesome. And, I think another thing that people might not think about so much is our earlier SARS issues back in 2002 and 2003, I think. Our first round with sort of a global SARS. It wasn't a pandemic, I think it was an epidemic, right? But we learned a lot about these viruses that really helped inform us moving forward. It's really exciting, amazing work.
Absolutely.
And then, thank you. And then I think, Katie, you were busy in your first year or second year of graduate school?
When
When this first started. second year?
No, it was my third
Oh, third. Okay. Sorry.
year. Yeah.
And so she was busy working away on a project to do with HIV when this happened. And like many, many graduate students, I think she got pulled away to get re-engaged and sort of refocus on figuring out what was happening with this virus. Would you mind telling us a little bit about what work you were involved in at that time?
Yeah, of course. So it was really awesome. So when we first got shut down, obviously, I was stuck at home for probably like the first week trying to figure out a computational angle to my thesis research. So I very quickly volunteered my services to Rick in the lab, and he immediately got basically a CV from me and approval. So I was like one of the first people that was actually from our lab allowed back onto campus to do some of the work. So I was really in the lab before the lab really had a very good idea of what projects we wanted to do. So it was a little bit scatterbrained at the beginning, to be honest. So a lot of it was just focused on me learning how to basically work with the patient samples. So at the time we were taking samples from patients in the hospital of the University of Pennsylvania. From, their oropharyngeal samples, so like back of the throat, nasopharyngeal, back of the nose, basically. And then we were also taking even endotracheal aspirates from people who were intubated, so we could really get samples deep within the respiratory tract. So I was trained up on basically how to process those samples and biobank them later, because Penn's basically started up a microbiome study, so looking at how the lung microbiome is affected by SARS-CoV-2 and whether or not that community can affect pathogenesis. So I just helped basically bank a lot of the samples for that study. And then I also helped isolate a lot of RNA from patient samples so that the lab could do genomic surveillance, which is a huge part of what the lab is currently involved in now. And then at the same time started the process of undergoing BSL-3 training to hopefully make some basically clones of the virus to really figure out how these variants are affecting like pathogenesis and replication in different cell lines. Yeah.
That's fascinating. And talk about a hot topic right now, right?
Yeah, definitely. There's a lot going on with the variants now.
You guys need to get together and figure out how the vaccines and the variants work together.
Yeah.
Pretty exciting. Thank you, Katie.
Mm-hmm.
And Callie. Callie had this unique experience. She's at Boston University as a graduate student again, and she's been trained in biosafety level 4. And what that means is when you see on the news and people working with Ebola with basically a spacesuit looking outfit, that's what Callie had been trained to do, because her PhD project is focused on Ebola. And so she was certainly already well trained and poised and ready to go. And I think it's many months of training, right, Callie? To get certification.
I actually finished my training during the pandemic. So you have different stages of training that you go through. And basically what you need to do is log 100 hours in the lab in order to get what they call independent access. But in the latter 50, as long as you know what you're doing and your mentor who's training you has approved you, you just have to make sure that you're in with somebody else who is approved. And at that point, you can independently operate your experiments on your own. You just have to be in there with somebody else, just in case,
Something happens.
You have a question on an SOP or something like that.
Okay.
Yeah.
And so then you were poised and ready to go and had all the training you needed in order to work with this new agent, which is technically one stage lower.
One stage below, yeah. So we actually got special permission both from the CDC and Boston Public Health to operate with the virus at BSL-4, largely because BSL-3 and BSL-4 training is quite different. The way you act in the hood, your protective hood is the same, but what you wear and the decon procedures and everything like that is different. So we didn't want to worry about having to train at BSL-3, and we're given access, or they allowed us to work with it at BSL-4.
That's exciting. And so what were you involved with at the time? What
So
kind of work are you doing?
I was actually personally coming off a high throughput sRNA screen that I was doing with Ebola virus. And basically what my lab started back in March was high throughput drug screening for SARS-CoV-2. So looking at drug libraries that we already have, largely focused on a library that contained already FDA approved drugs, as well as novel drug candidates to see if we could find anything that could stop the virus from replicating in cells inside using the drug. So just normal cell infections. Aside from that, I played several supportive roles. In the early days, we didn't really know how to purify the virus.
Yeah.
So I got to really kind of figure out what sort of, basically what sort of sugar polymers worked best for concentrating and getting a pure stock. I developed a high throughput RNA FISH technique for detecting the virus. So we could actually add basically virus replication, looking at genomic transcription as a step to screen for in our drug screening. I should probably mention my lab uses an image-based platform. So basically what we do is we infect cells with a virus, and then we add different agents to essentially illuminate the virus within the cells and see how many cells got infected in a sample. So I actually use that technique to develop a way to really look at a particular step of the infection cycle, specifically looking at transcription. And with that technique, along with other techniques in the lab, helped to participate in screening or looking through our entire, basically all these different drug libraries we had. And aside from that, I also got to collaborate with a lot of different Boston researchers on different projects with SARS-CoV-2. So I got to collaborate on this really cool project looking at how we could possibly use remdesivir better. Most of you probably know that I think remdesivir is the only FDA approved drug for SARS-CoV-2 right now. One of the problems is that we can't use that drug far enough to get a bigger effect in terms of treatment. So this project was actually looking at what can we do, what else can we give remdesivir with in terms of other drugs in order to allow us to treat patients with greater amounts of remdesivir and thus let it be more effective against SARS-CoV-2. So that project was really focused on if we knock out certain genes within a cell using CRISPR technology, how much remdesivir can they stand? And with those gene knockouts, do they still process remdesivir the same? And does remdesivir still work against SARS-CoV-2 when these particular genes were knocked out? So kind of getting at this idea of, can we administer remdesivir as a drug cocktail?
Wow. Well, you guys are amazing. It's incredible what you're off doing, and so right center in the middle of all of it. It's really impressive. I'm extremely, extremely proud of all of you. It's incredible to hear all the incredible things that you're doing, and the implications of your research is so important moving forward. It's really incredible. And so I was wondering if you had any personal stories of what it was like. I mean, I know Katie shared with us, I think that's incredible to be one of the first people back, on campus. I know I had a similar experience because I was doing a COVID-related project, and I would come, and literally my car was the only car in the parking lot, which is sort of bizarre. And so Katie, I know you were sort of like most of us, right? Going home, what can I do computationally? Not much. Okay, what else am I going to do? So, it's pretty amazing, your story. Were you in lab alone at that time, or were other people there?
There was one other person. So Rick has a staff scientist who's been with him longer than I've been alive.
Okay.
And he was the first person back. I don't think he ever actually left. Probably. And then I followed very shortly after that. But yeah, it was definitely very strange. We had to carry around letters to show to public safety officers to prove that, we were essential personnel. We weren't just trying to sneak in to conduct some sneaky research on our thesis projects.
Which I'm sure happened.
Yeah, under the table, probably.
Yeah. And Alec, what was Manhattan like at the time?
It was like a scene out of the movie. So I was right next to Central Park, and they were actually building this field hospital. And maybe you heard in the news, this was in March, right when it was hitting New York hard and the lockdown went into place. They had actually large hospital ships on either river just taking the overflow, and you go outside and New York was deserted, which never happens. It was terrifying, and we were brought back into the lab to work on COVID, and the lab is right above the hospital, so you're constantly walking through and just thinking about patient contact. And I had a very good friend who was pulled into, he was a researcher but had his MD, and they pulled him into the clinic because they just didn't have enough staff, and staff were getting infected. And he was telling me war stories. It was a very, very challenging time.
Surreal. And
Yeah.
Callie? Boston must have been strange too, empty.
Yeah, so what was interesting for me in particular is inside the lab, nothing changed for me because the lab, my lab on my floor and the other labs on my floor, we all had our COVID protocol or SARS-CoV-2 protocols approved just as they were shutting down all research labs at Boston University. So we never actually were required to shut down because basically it was a few days difference of getting those protocols approved, getting a sample of the virus, and getting going in the lab. Now, like Alec, we're right on the same street as Boston Medical Center, which is known for treating many, many patients with a specialty towards treating low-income homeless patients as well. So we had, the street itself never really slowed down.
Yeah.
If you went a few blocks back to like the residential South End area, it was dead quiet. And that was really, really strange. I started carpooling in instead of taking public transit. Now I bike in because my carpool moved. But driving in, I think, was also very odd. If anybody's ever visited Boston, you don't really drive in Boston. You just kind of pray. So it was very strange seeing the empty streets, being in the car, being able to park on the first level of the parking garage sometimes. That was all very, very odd. So yeah, I guess that was my experience, just kind of seeing everything suddenly outside turn off.
Yeah, absolutely. It's incredible. And I know we have a few current students who are probably eager to hear about how did you get involved in undergraduate research while we're at RIT. And I know I kind of stole Katie for her entire time at RIT, but I know Alec and Callie were also involved with other faculty members. And so I was wondering if you could just talk a little bit about why you wanted to maybe do undergraduate research, how you got involved, and if you felt like that helped you maneuver through to the next stages. And you can just go.
Okay, I'll go, I guess.
Go for it.
Yeah. So when I originally came to RIT, I knew that I liked science, but I wasn't sure if I wanted to go like the medical path and go the MD route, or if I wanted to get involved in research. So I actually did both. So I joined RIT Ambulance, did that, worked as an EMT there for like 4 years. And then I think in my like first semester is when I first emailed Dr. Ferran, and I actually wasn't even interested in viruses at the time. I thought I was interested in cancer biology, but she gave just such an amazing talk at one of the slide slams put out by the College of Science. I don't know if that's still an active program for students, but basically it was just the faculty giving like 5 to 10 minute speeches about their research, and her stuff was so cool. So I sent her an email and she luckily took me on.
No, luckily for me.
Sure. So that's how I got involved in that. And I spent 4 years in her lab. And then I knew I liked the research angle at that point. I also enjoyed my time as working as an EMT. So one of the reasons why I wasn't in graduate school earlier was because I then went to the NIH because of those like experiences and tried to get involved in really, really translational research. So I was working there with like rhesus macaques, and we were looking at the different microbiomes within the vaginal tract from human patients. So it was a really cool opportunity to do that. And I definitely would not have been accepted to that if I didn't have the 4 years of undergrad experience that I had at RIT. And then that experience was a huge plus for me when I applied to graduate school. Then 2 years later, I've had people tell me that, you know, you're here because of your experience at the NIH. So it's all been like a chain of events for me, of how I ended up where I am. And I'm glad that now I've kind of come back to my original scientific roots in virology and some microbiology now.
Yeah, I used to laugh when Katie was in the lab. I said, I think Katie's the student who doesn't sleep. Pretty much. Alec, what about your path?
Yeah. So I started at RIT, and at the end of the year, I wanted to get involved in research over the summer. And I also, like Katie, had no idea what I wanted to do. And I talked to Dr. Sweet actually to do developmental biology. And so I got involved with her research, looking at early developmental phases of neuronal systems between different species of sea urchins, which is incredibly fascinating. We did some amazing work, and Dr. Sweet was such a phenomenal mentor, helping me learn to do science and to learn how to write science. And those early trainings really set me up to do well later on. And then, unfortunately for Dr. Sweet, I took Dr. Ferran's class in virology. Maybe I shouldn't say unfortunately. And I fell in love with viruses. And so I had to make the toughest decision, which was to switch labs and to join Dr. Ferran's lab for 2 years at the end of my undergraduate and start diving into viruses. And since then, throughout that research, I learned how to do cell culture and manipulate viruses and titer viruses and grow them, count them. And all of those skills translated directly into what things I did in graduate school. And when I was interviewing for graduate school, I remember I was deciding what to do at the end of undergrad, and I went to Dr. Ferran, and she's like, "Oh, you should do a Ph.D." And I'm like, okay. She was like, "You should check out the school Mount Sinai. They're great." And so I'm like, all right. And I applied and went there, and it was just a bunch of people very, very passionate about virology. And I told them the work that I'd done and the interest that I had, and they were ecstatic to hear someone talk about viruses and virology in such a way. And so I was very, very grateful to be accepted there. So it's just a lot of stumbling and some good luck and some amazing people.
It's really true, isn't it? Life just takes you spend all this time worrying. And then, so I was at a conference, American Society of Virology, about your first or second year of graduate school. And I happened to sit on a bus next to a faculty member at Mount Sinai. And I said, "Oh, do you know Alec Freyn?" He's like, "Oh, he didn't join my lab. I'm so depressed." He was so great. I was like, "Yeah." And you know, I think that's something you touched on that's really important, because so many students, I know Callie, Katie, and Alec, you know, we had so many conversations towards the end of your time here, and deciding which school to go to is very, very difficult because you're narrowing your path at that point. And so I think it's really great. I think I always I told you all, I think, right? There's no such thing as a bad decision, especially in all of your cases, but it's not an easy decision at all. And then Callie, what about you?
Yeah, so I came to RIT knowing I wanted to do research because my senior year of high school did a really fun science fair project that was really the highlight of my senior year. So day one of intro bio, I walked up to my professor, Dr. Dina Newman, and I said, hey, how do I get involved in research at RIT? And I actually did 2 labs, I think, the entire time I was at RIT. I was part of her group studying biology education, but she was on that first day mentioned you, Dr. Ferran, and said that you were looking for students and that you would take freshmen, which was really exciting for me because I was a freshman. And so I emailed you, and luckily you took me on. And I learned like right off the bat a bunch of critical skills. Katie actually taught me tissue culture in your lab, which was super cool. And I was in your lab for those first 2 years, loving it. But like Katie, I thought I came in wanting to do cancer research. I didn't really quite want to abandon that initial idea. So junior year, I switched into Irene Evans' lab to look at organoids and drug development inside the organoids, and was also taking biology of cancers. And as cool as the work was, I realized I really, really missed viruses a ton. And so that summer, I actually applied for an internship that Alec had actually initially been the one to tell me about. I don't know if you remember, at the Pasteur Institute in Paris. And by some stroke of luck was accepted into it. And there I actually kind of got to marry the 2 research ideas, where I was studying the cell biology of HIV entry across the nucleus. And I absolutely loved it. Came back to RIT in the fall, took virology with Dr. Ferran, and kind of rejoined your lab in that last year because I just missed the viruses so much. Applied to grad school, ended up choosing, after many conversations with you, Boston University. Came here actually expecting to do HIV work, but rotated in an Ebola virus lab that studied the cell base that had a lot of high throughput aspects to it, which were really fascinating, but also really studied the cell factors that were important in allowing the Ebola virus to replicate in cells. And that was kind of like the poison that caught me. I was just so fascinated with that. So that's really how I got here. And getting into grad school and being able to talk about research and viruses the way I did was really a product of being in your class and being in the classes at RIT and talking about science like we did at RIT. The educational research aspect helped a lot as well, because I learned how to communicate better. Just those skills, the buildup of those skills in the research labs and in the courses, the directed research courses especially, were critical, I think, to helping me score the opportunities I was able to score and get into grad school.
Well, I think you're all amazing, and I love how humble you are. It's incredible. It's incredible. And so I think it's close to 6:40. I'm happy to open it up. There's a Q&A. I'm going to put my glasses on here. All right, so we have some questions. Is there anything special about COVID that makes it more susceptible to an mRNA vaccine? That is, why have the COVID vaccines been apparently so successful where vaccines against other viral targets have been failures? That's a political question, right? Well, there's a political answer. Or is it the delivery mechanism that Alec described that made it all work? That's a great question.
That's a phenomenal question. I think I could tackle just a small part of that. The mRNA, one thing is the virus. The virus has just one target on the outside of that we can use as a vaccine target. And it seems that vaccines that make good antibodies against that target are very effective. And so this is kind of the classic way that vaccines function. So things like the measles, mumps, rubella vaccine, things like diphtheria, tetanus, pertussis, all these different vaccines function mostly through eliciting neutralizing antibodies, so antibodies that block the virus from getting into cells. And so luckily, this isn't a very strange virus. If we can elicit these antibodies, we can protect. And one of the reasons potentially why the mRNA vaccines, both Moderna and Pfizer's vaccines, are so effective is that they are able to make that piece of the virus directly in your body. And so one of the issues is that this piece of the virus, the spike protein, tends to fall apart or be unstable. And so if you keep it for long periods of time, even the protein can be relatively unstable. So because the mRNA immediately makes the protein in your body, there's less chance for the protein to degrade. So that's one of the potential reasons that the mRNA vaccines seem to work very well. Other vaccines are also incredibly effective, such as Johnson & Johnson's vaccine works incredibly well to general vaccine standards, as well as Novavax is coming out with vaccines, and AstraZeneca. All of these vaccines comparatively to other vaccines throughout the years are phenomenal.
Yeah, no, I think that's exactly true. And, as you know, right, I believe you were primarily developing these mRNA vaccines against flu, right? And so I think I said it was political because I think it was in the works for other viruses. And then a pandemic came along, and thankfully we were able to switch targets, I think, pretty rapidly because of the technology. And now with the vaccines, they also hope that they can switch those targets up so that the new vaccines can equally target the variants like Katie was talking about. So it's one of those cases bit like HIV, right? We totally changed our regulatory rules and our approach to accommodate a really problematic virus. And we see that, playing out here, which is really interesting.
Mm-hmm.
Okay, how does remdesivir work? Katie, Callie, you want to give that a try?
Yeah, so remdesivir is what's called a nucleoside analog. So basically what that is it's essentially like a base of RNA. However, it's slightly different. And I believe the way remdesivir works, correct me if I'm wrong, if any else remembers, is that it actually stops. The way it incorporates into the developing RNA strand is it actually stops further synthesis off that strand. So essentially it gets in the way of the RNA polymerase, the thing that makes more RNA, and it mucks it up, it causes it to stall, and it just prevents the virus from replicating.
That sounds exactly it. Yup. Let's see. What factors, oh, that's a great question. What factors did you take into account when selecting a graduate school? Katie, every single thing under the sun.
Yeah. And Dr. Ferran knows that because I sent her many emails about it. Even I was 2 years removed from her lab at that point. And
I'm always here for you.
Yeah, thank you. I appreciate that. Yeah, I had a devil of a time selecting a graduate school, mostly because I was fortunate enough to get accepted to a lot of really cool places.
Yeah. Okay. Fortunate enough, amazing enough to get into a lot of places.
At the end, it really came down between two schools for me. It was the University of California, San Francisco, and the University of Pennsylvania. They do amazing science at both. That wasn't really a question. I knew I would have liked the science at both schools. They both do amazing caliber research, all that. What really came down for me at the end, deciding between those two schools, was that UPenn has they just have a ton of virology faculty. There's 40, virologists. And sort of when you go to grad school, I think everyone has an idea of, oh, I want to do this. And, I want to work in XYZ people's labs. But a lot of the times that's not the lab that you end up joining. I actually came to Penn thinking oh, I want to work on a virus, but I definitely don't want to do HIV. And now I find myself in the middle of an HIV thesis project.
And Callie wanted to work on HIV, and now she's working on Ebola.
Yeah, exactly. So I chose there because there are just so many options, and it feels sort of more like there's a more family sort of environment. There's a lot of people around you who get really excited about viruses, and I like to sort of feed off of that attitude. So that's how I ended up selecting there, and I have zero regrets. So yeah.
And Alec?
Yeah, I think it was also an incredibly difficult decision. I think when I visited Mount Sinai, the virology department is really on 2 floors, and it's very open door and very collaborative. And what I enjoyed was people there. It's a very hardworking environment, but it's also a very helpful environment in terms of everyone is willing to lend a hand, let you borrow a reagent, let you talk about just troubleshooting science in general. And I just really enjoyed that aspect of the feel of the department. It was a very close knit, and just a lot of, like Katie said, just a lot of people very, very passionate about viruses. And it's easy to get swept up in that passion, and easy to get pulled into such a fast-paced, high output environment, which was just, it was an incredible place to do graduate school.
Yeah, there's something about having a critical mass of people all interested in something. It can be different viruses, but the questions are often similar. That's just very, very invigorating and exciting. And Callie, I remember you've talked a little bit about it already, but you had a heck of a time deciding.
Yeah, so I was lucky to have a few offers, and I should preface this by saying I have difficulty making decisions anyways, but all of the places were fantastic. I had done some pre-filtering in where I had applied based on location. I would have loved to apply to Mount Sinai, but I also knew I could not live in New York City. So like that was essentially my first cut, is I didn't apply to places that I didn't think I could live in.
That's an excellent, excellent point to consider.
Yeah, especially currently with the pandemic, people can't travel to the cities that their universities are in. So like having that background knowledge was really key. Aside from that, evidently flew out. I got to fly out and go to the schools, meet the students and everything like that. And for me, it really came down to both kind of the happiness and excitability of the students, and also just the diverse research ideas and resources that were available. And really why I chose to come to Boston University was during my interview, all of the students I talked to were super excited to talk about their research. And, I wanted to be in grad school and like the hard, days that make you want to like go crawl into bed and never come out again, and remember why I had, put myself there. And so loving what you do, like for me, it was really just it was about being able to love what I did research wise. And I'm here and I love what I do. And I loved my department. Again, family group was really important. You could see that from the interview, and my department's kind of like my second family. That aspect was really key and critical.
Yeah, no, I remember you were almost trying to talk yourself out of it, if I remember.
Yeah.
So I feel like we touched on this a little bit, but somebody was wondering how your undergraduate research influenced your career. If anybody has anything they want to add to what they've already said, I feel like we've touched on that already, but. No.
I can add one thing. You would give us primary literature articles to read a lot. I really like the fact that you sent us out papers to read and stuff like that. And I always think back to, like the first year I was in your lab when I could barely get through a paper and hope to understand maybe 10% of it. You gave us a paper about the RVSV Ebola virus vaccine. And that was something I didn't realize that I'd be working on Ebola virus later on, but the concept of that paper just hooked me on to the idea of studying viruses, because I was so fascinated by the fact that you could take a protein from one virus and stick it on another and make the body recognize it as that virus, which I thought was super cool. So can add that into primary literature within the undergraduate research was really key.
Yeah, it's a tough nut to crack.
Yeah.
Oh, this is a good question. Did any of you get COVID?
Yeah, so actually the weekend before the lockdown, my significant other went and was going to a wedding just across the border in Jersey. And she was like, I don't know if I should go. I don't know. And it was that kind of era right before where all the hesitancy was going on. And she came back and we both got COVID. And it was it felt like I was lucky that it was relatively mild and felt mostly just like a fever or a flu for a few days. And then with that, my antibodies were actually a very high titer, so I was able to donate some plasma for the initial plasma transfer experiments and use that plasma or serum also as a positive control for some assay development. So it was useful as well. But it's yeah.
No
very-
fun. That's funny. Oh, well, I'm glad you're better.
Yeah.
And Callie, you mentioned that you worked in different labs as an undergraduate. Is that something that you would recommend other students do, or should they stay in one lab? This is a great question. I think all of you come at this question from different perspectives.
I think exploring more early on could be advantageous, maybe like within the first 2 years. Unfortunately, you can't really do lab rotations as an undergrad, but kind of feeling out those different areas. I think one of the things I regret the most is, especially during junior and senior year, when I was at a point with my education at RIT where I could understand more advanced material, not having more time with you in your lab, and having those opportunities, I guess, just to get more work done and get cooler things done. But I think being able to try out multiple things is really important to being able to figure out what you like and what you don't like. I would just suggest trying to do it if it's possible within the first few years. That's my take.
That's a good answer.
Yeah, I think it was very, very helpful as well to know. And I think just getting involved, if you think you want to do research, just getting involved early, because having that relationship with a mentor who can really teach you how to think about the research questions that you want to address and how to write science. And I just really appreciated that, especially from Dr. Sweet getting me started, and then Dr. Ferran from stealing me away. And it's just I think you learn so much regardless of the topic. And then secondary, you get to learn what you are really passionate about.
Yeah, and I think also, different mentors, and this you see in graduate school too, right? Different labs have different personalities, and outside of even the science, you can work with somebody and you could love the science, but there might not be a really great interpersonal relationship there, or, there might be some issues sometimes. So it's always good, I think, to try to reach out to different people. And Katie, you kind of did it the opposite, right?
Yeah, I did.
4 years with me.
I ended up
after.
Exactly. Yeah. I just had too much of a good time in your lab, and we clicked. So I had no interest to really join another lab while I was there, right? But at the end, after I graduated, I was like, well, I ended up in this virology lab. I didn't think I originally wanted to do virology. Maybe I just like science, like just solving those puzzles in the lab. So that was when I was like, let me just try something totally different. So at the NIH, my first project was working with rhesus macaques, and we were working with like the concept of whether or not there is a small population of cells in an adult ovary that can actually give rise to new oocytes, which is sort of contrary to the dogma that's been around since like the early 1900s. Obviously a very different project. I got to learn a lot of really cool like translational skills. I got to learn how to suture monkeys, help do some laparotomies, read some ultrasounds. So all of that was really cool. And then I ended up in a Drosophila oogenesis lab studying spindle formation. So I really ran the whole gamut. And it was while I was, and while it was away from those things, or when I was away from virology, that's when I sort of recognized like, wow, I really miss like the virology research that I was doing. And I wasn't around virology people, obviously, when I was working at the NIH. And so when I came back to grad school, I knew very firmly that I wanted to stay in virology. And I might have always had that lingering doubt if I hadn't gone and done those other two things. So it's probably advantageous if you do it earlier, but if you do it later, what's an extra 2 years on top of the 5 that it takes you to get a PhD? It's like not that much. And definitely having that diverse skill set is very attractive to new labs and labs that you're looking to join. So yeah, I still recommend doing that.
You guys are amazing. Why do mRNA vaccines need to be kept at -80?
Yeah, that's definitely a good one, especially.
Or -20?
So the reason is that little bubble that I was talking about before, that lipid nanoparticle and the mRNA itself, both of these molecules are prone to degrade or fall apart. So basically, if you keep it really, really cold at -80 C, you stabilize the molecule just by forming it into a compact frozen solid. And this just prolongs the shelf life of the RNA. If you leave it at room temp, basically that little fat, it's kind of like butter. You leave it at room temp, that little fat bubble melts, and then everything is destroyed.
Well, you have to figure out how to stabilize it, which I'm sure Moderna is working on, unless I don't understand anything. Alec, how many epitopes does the Moderna mRNA vaccine target?
Yeah, so.
Are you allowed to tell us?
Yeah, so right now it's just targeting the spike protein. Within that protein, there's multiple epitopes that have been determined by other laboratory groups, specifically groups such as James Crow and others. So basically, little regions of the protein, you can think of it kind of like a stalk, like a flower, kind of a stalk and a head. And there's different regions that are more attractive to the B cells, which are making your antibodies. And so there's certain regions such as the receptor binding domain that is what the protein engages with to get into your cells. And that region seems to be very immunogenic. So it seems to stimulate antibodies very well. And there's also a region called the N-terminal domain, which is kind of a scaffold underneath that receptor binding domain. And antibodies to those regions also seem to neutralize quite potently. And those antibodies are not, to our knowledge, they don't seem to reduce neutralization titer as much to some of these variants, depending on the variant. So in terms of the target, it's just the spike protein. And then within the spike protein, there's multiple epitopes that are targeted.
Fascinating. How do you recommend doing something like research in between undergraduate and graduate school? So like taking a gap year, perhaps?
Guess it depends on what you do with the gap year. I don't think it'll hurt you to take it. I mean, I took 2 gap years. It certainly didn't hurt me, other than me being one of the older people in the program. I think in general, grad schools tend to sort of like that stuff, like you to have like unique experiences like that. Or if you go and pursue research after undergrad, then that's really showing like a level of commitment, right? Because it requires a lot of commitment to get through grad school. So if you willingly like take time off to do more research, then they're very sure that you'll make it through the program. So it's up to you. There are certainly graduate students in the UPenn program who have come straight from undergrad. Both paths are fine. It's just up to you and sort of the opportunities that you take along the way. If you didn't have a lot of undergraduate research, if you maybe got involved in it late, then it's certainly better to do something in between undergrad and graduate school. But if you have a really awesome experience and you've been doing like summer undergraduate research and stuff like that, then you can probably get in without it.
Yeah, that was asked by one of my current research students who've been with me for 3.5 years. So, hi, 2 questions. When will we see, oh, this is a good question. When will we see the end of the pandemic, and how do we move forward in the wake of these new SARS-CoV-2 variants? The $10 million questions.
Yeah, I don't know if I can even speculate on a timeline, but I can talk a little bit about the variants, since those are things that we're actively tracking in Philadelphia. And I know there's a lot of interest in like tracking the UK and the South African variant. So I can tell you, at least for the Philadelphia area, that we have started to see actually the UK variant circulating here. Some of them have come from students who have recently traveled there, and others have come from community transmissions. So at least in Philadelphia, we are having local transmissions of that variant. We also have local Philadelphia strains that have some of the same mutations that the international variants have. So, you know, the UK variant and the South African variant both have mutations in the receptor binding domain. And some of those, you know, are thought to be linked to increased transmissibility. And it may be affecting basically antibody recognition of some of those epitopes. So that's definitely stuff that we're very worried about as the pandemic progresses. And that's one of the reasons why we're tracking it. Unfortunately, we just don't really have enough data right now to really figure out what a good clinical correlate of vaccine protection is. So like we can take a variant and say, you know, if they have this mutation, and you basically make that virus and you incubate it with an antibody, you can show, you know, maybe you get decreased binding. But that doesn't really say anything about whether or not that vaccine can actually protect you in vivo, because there's more things that are happening with the immune system than just that one mechanism, even though that's a lot what the news is really focusing on right now. So I think it's really important to like distinguish those two things. We really need a lot of data, and we need data from geographic regions that have a high proportion of that circulating variant. And then you need to really compare what the infection rates are and the transmission rates are as you're sort of vaccinating people, and compare that to unvaccinated controls. And that will really give you an idea of whether or not that vaccine is protective against the variant. And unfortunately, we just don't have that data yet.
And the big thing, right? Everybody stay home as much as possible.
Yeah.
Everybody needs to get vaccinated so that the virus can't replicate and these variants don't arise, right? So let's try to keep things under control a little bit longer. Just put our head down and keep the end goal in mind.
I agree with everything Katie's saying. And I just want to inject a little bit of optimism in terms of the vaccine rollout. I've been obsessively tracking the Bloomberg and the New York Times vaccine rates, and we seem to be just increasing in terms of vaccination rate. So on Saturday, actually, there were 4.6 million vaccines given out in the US. So it's very encouraging to see more and more people get vaccinated, especially now seeing the tail end of high-risk groups being vaccinated is incredibly nice to see. And to see the cases keep decreasing and the deaths keep decreasing per day, it gives me a sense of optimism. I'm worried, of course, about the variants, and I'm want to stay realistic, but I think it's good to cling on to some hope.
Oh, absolutely. I agree. But I read this morning, 1 in 5 Americans have been at least one dose of a vaccine, which is an extraordinary feat. I mean, impossible
3 months.
to imagine. I don't know if we have time for one more question, Bill. Okay, so do you get certified for BSL 4? So this would be for Callie. Oh, you went away. I think the question was about, do you get certified for BSL 4 through the CDC? How does that process work?
Yeah, that's a great question. It's slightly dependent on where you are and what you're doing. So every institute in the United States, you will get certified eventually through the CDC. Being in Boston, we also have to get certified through the Boston Public Health Commission. We have an extra safety committee we have to pass. But the initial process starts with essentially a very intensive Department of Justice clearance, so 10-year background check, lots of forms you have to fill out, and then you have to get medical clearance in order to be like medically certified, your sound and everything like that to go into the lab. You have to get clearance in the lab. So I had to have references from professors, and I think Gabby Crandall in BioPrep wrote me one as well, for basically to attribute like my lab work and say that, yes, this person can work safely at BSL-2, this person can be entrusted to work in this environment. In addition to that, I had to be cleared with a shadow, essentially like a non-formal shadowing program in my lab. My PI had to essentially stand over my shoulder and watch me work at our BSL-2 to make sure that I was not making any mistakes and stuff like that. And then that went forward to our institutional biosafety committee and training advisory committee, and they approved me to begin training and to begin submitting all of my paperwork and everything else to the governing body, so the CDC, the FBI for my background checks and everything like that. So it was a slightly long process. I think we started the clearance and the paperwork in May. And my first day inside the BSL-4 was in August. So it wasn't terribly long. The thing that actually took longest was waiting on Boston Public Health. Believe it or not, it took a little bit longer than the CDC. Yeah, you have to have patience. You have to be okay with being watched in the lab, having somebody stand over your shoulder and watch what you do and make sure that you're not making mistakes and correct your mistakes when you do make them. But yeah, I mean, once you get through it, it's a skill that you have, and it's pretty cool. Yeah.
Well, I know Bill has some concluding remarks, but I just want to really, really, from the bottom of my heart, thank all three of you for participating. I think it's invaluable for our current students and also alumni who, your career's never done, right? Even in my old age, I'm always learning new things and taking on new directions. And so I really thank you for sharing with us your experiences. And I'm just so thrilled to have been a part of your path. And I thank you very, very much. You're just all incredibly amazing people and scientists.
And myself, I would also like to share that vote of thanks to all of you, including you, Maureen, as well. Maureen, Kaitlin, Callie, and Alec, just for your time tonight and just describing how influential RIT was for setting you on your path and pursuing your passions, and being a help to all of us throughout the world, through your work in research, whether it's therapeutics, testing, vaccines. It's amazing, and it's amazing to see what comes out of RIT, and I think tonight's a perfect example of that. So before we leave, Kayla, my coworker, is actually going to share a link for another event we have coming up next week, March 24th, bioinformatics, utilization of artificial intelligence in medicine. So that'll be at 12 p.m. on March 24th. Feeling a lot of people viewing tonight might be interested in that webinar as well. So be sure to tune in for that. I will send out a follow-up email with that event as well as a recording from tonight. And for any students that are on, I'm sure Alec, Callie, and Kaitlin would be happy to connect with any of you if you wanted to connect with them offline and just chat one-on-one about their experiences and your path going forward. So I'll be sure to share that as well. But once again, thank you all for joining us here tonight, all four of you. RIT Tigers, be well and be safe. Thank you all.
Thank you.
Take
Keep
care.
in touch,
Bye.
you guys.
All right.
Bye.
Thanks.